Hello,
Rob.
this is Robert Cothy with the EPA Region 1 office. I'm going to be moderator for today's session. Welcome to our program entitled Tick Threats and Controls, a panel discussion for camps and recreational land managers. Today, we'll hear from experts on ticks, the health risks ticks pose, and we'll learn about how to protect ourselves from tick bites through personal actions and integrated pest management. Before we begin our panel discussion, I have the honor of introducing EPA's Assistant Administrator for the Office of Chemical Safety and Pollution Prevention, Alexandra Dunn.
Thank you so much, Robert. It's great to be with you all this afternoon. And before I begin my remarks, just want to wish everyone on today's webinar the best of health in this unprecedented time. Also, want to say thank you to all of you for your efforts during the pandemic. As managers of our outdoor recreation areas, we know that you are always on the front lines of issues like tick safety and mosquito control. And now you're facing a new suite of public health demands at a time when more and more Americans are turning to their local parks and campsites for vacations, picnics, and a breath of fresh air outside the home. So again, thank you for all you do. And with that in mind, I'm really glad that we are able to come together today for this webinar. Just as people head out for summer vacations, it's really important to focus on managing risks from ticks. I'm also happy to be on this webinar with my colleagues from Region 1. Some of you may know that in 2018, I had the privilege of serving as Regional Administrator for EPA New England, and I was able to work with the experts on today's call and many of the staff. And I saw firsthand what Regional Administrator Diesel will be speaking to in a moment, which is the immediate need for action on tick-borne and mosquito-borne illnesses. When Dennis and I passed the baton between each other, he told me he really wanted to work on something regarding tick and mosquito-borne illnesses. And I'm really glad that we have this chance to work together. So thank you for joining us for the first webinar in a two-part series. We hope that this series will be relevant to you because we created it to support recreational land managers, public health professionals, and other outdoor recreation sector stakeholders who are working hard to protect the public from tick and mosquito-borne illnesses. As you know, today's webinar, we will be hearing from a panel of experts who will provide national and local perspectives on ticks, the diseases they transmit, and methods for control. And our second webinar, which is scheduled for July 16th, just next week, will take a closer look at mosquito threats and control measures. And if you haven't already registered for next week's webinar, I really encourage you to do so. It will be held again next week at the same time as this webinar. Just in closing, at EPA's Office of Chemical Safety and Pollution Prevention, we receive questions and concerns on these issues from the members of the public, businesses, state and local governments, and tribal leaders nationwide. And dangers from ticks and mosquitoes are present in all corners of the country, but every region of our nation has a unique set of local threat vectors, a different set of management needs, and locally driven public health protection requirements. That's why, again, we were glad to bring these two webinars together and take a deep dive into New England specific rules, resources, and approaches that hopefully will help you keep open spaces safe. And beyond the strategies that you'll be hearing from on the panel, I do want to mention briefly that our office at the EPA headquarters is working hard to help you address other risks presented by the coronavirus public health emergency. Our pesticide program is responsible for what's become known as List N, as in Nancy. That is the list of disinfectants that are approved and known to be effective for use against COVID-19. And we've even built a web platform that's mobile phone friendly to make searching List N more straightforward and accessible to everyone. We've worked with CDC to provide guidance on how to properly clean and disinfect public spaces, such as the areas that you may be responsible for as attendees on this webinar. And we will provide links to these resources at the end of the webinar. So in closing, even in the backdrop of the pandemic, the challenges associated with tick-borne illnesses have not subsided and cannot be overstated. As we know, New England is a place where tick-transmitted diseases continue to increase, and everyone is very aware of this concern. We are here to help. Our pesticide program team regulates tick control products. We work with our states, and we collaborate with national experts to advance understanding of ticks and tick management. And we're so glad to have so many of them on today's webinar. Please, after today's webinar, if there's anything you think that the US EPA can do better to protect you, please feel free to reach out to me or to Regional Administrator Dennis Diesel. And with that, I have the great honor and pleasure to turn the podium, the virtual podium, over to New England's Regional Administrator Dennis Diesel.
Thank you, Alex. Thanks for the virtual podium. And hello, everyone. Thank you, Alex, for joining us today. It's really an honor to be here with you. Again, my name is Dennis Diesel, Regional Administrator for EPA New England. I would like to start by echoing what Alex said. Just in the middle of the pandemic, I hope everyone has had a safe, healthy spring. And as offices begin to reopen and families scramble to create new vacation plans, I do hope that everyone is staying safe at work and at home. And despite facing these unprecedented operational, personal, professional challenges, I am grateful to everyone on the call today and our entire community of partners throughout New England for your efforts to protect public health from the pandemic and from ticks. As a native New Englander, I'm from Goffstown, New Hampshire, for all those Goffstown residents, a lot of pine trees in Goffstown. I certainly understand and appreciate the challenges facing all of us as we work to balance the benefits of time spent outdoors with the risk of exposure to tick-borne disease. By now, most of us know that New England is a hotbed for Lyme disease, but that is just one of several tick-associated diseases that pose significant public health concerns across the United States. This hard truth has been affecting New Englanders for some decades now, and it's been affecting more people with more diseases and more fields, farms, and forests every year. As Alex noted, even during a global pandemic, communities in all corners of New England have a dire need for more resources and more tools to manage ticks. And I saw this firsthand. I've been regional administrator since last summer. When I came back to New England, my brother actually had Lyme disease. I went to a couple of Superfund sites, and we had to be especially careful for ticks. And then later in the fall, I went on a tribal visit up in Maine, northwest Maine, and they were telling me the story of they came upon a sick moose, and I didn't see the moose myself, but they sort of asked me to guess how many ticks they had found on the moose, and I guessed a couple hundred, and I was kind of horrified at the prospect of a couple hundred, and they said based on what they saw and some of the numbers they crunched, it was probably closer to 50,000, and so that was kind of a red flag for me that there is certainly a tick issue, and we need to address it as best possible. So as campgrounds and recreation areas begin to reopen and again welcome the public, we do hope you find today's information and resources on tick threats and control measures to be valuable and timely. As again, Alex walked through a little of this, but first we'll hear from a panel of national and local experts providing perspectives on ticks, the diseases they transmit, emerging trends, and methods for control. And then at the end of today's presentation, our panelists will be joined by representatives from our New England state pesticide programs to answer your questions about ticks and tick control. So with that, thank you again for joining us today. I'll turn the webinar back over to our moderator and a member of our EPA New England pesticide program, Rob Cosey. So Rob, back to you.
Thank you, Regional Administrator Diesel, and thank you, Assistant Administrator Dunn. As was said, the emphasis will be on New England pests, which the focus will be mostly on the black-legged tick, the deer tick, which is the tick that transmits Lyme disease and is probably the most serious problem in New England among the ticks, but other tick species issues and tick-borne diseases will be discussed as well. And as was said, the focus is on how to deal with tick problems in campgrounds and recreational areas. Before we start, here are a few tips on viewing the webinar. To toggle between a maximized and standard view of the presentation, click on the four-corner symbol highlighted with a yellow arrow. If you're having technical difficulties, press the raise your hand icon, which is highlighted with the blue arrow, and we'll do our best to help. To download a PDF version of the presentation, press the paper icon, which is highlighted in green. And then to pose a question, use the question box, which is shown with the red arrow. We're fortunate today to have three prominent scientists and researchers who have spent many years studying ticks and tick-borne diseases. Our panelists include Dr. C. Benjamin Beard, who is Deputy Director of the Division of Vector-Borne Diseases in the Centers for Disease Control and Prevention, Dr. Kirby Stafford, State entomologist and chief entomologist with the Connecticut Agricultural Experiment Station, and also a scientist with the CDC's Center for Vector Biology and Zoonotic Diseases, Northeast Regional Center for Excellence in Vector-Borne Diseases, and Dr. Stephen Rich, who's director of the Laboratory of Medical Zoology and a professor of microbiology at the University of Massachusetts, Amherst. Dr. Beard will begin with you.
Thanks, Rob. The talk I'll be giving today is tick-borne diseases in the United States, burdens and trends. And see, I don't think I have, there it is. Okay, so just in terms of letting you know where I'll be going in the next few minutes, first of all, I'll talk about the key tick-borne diseases and a little bit about their burden and distribution in the United States with emphasis on Region 1, the EPA Region 1 in New England. Then I'll talk a little bit about disease trends and drivers. What have we seen over the last couple of decades, and what are the drivers? What are behind these trends? And then finally, I'll close by saying just a little bit about tick-borne disease prevention and control. And I'll only touch on that because that will be covered a lot more thoroughly by the other two speakers. So in terms of the situation with vector-borne diseases in the United States, between 2004 and 2018, we had more than 760,000 cases that were reported in the United States to the US CDC. The number of annual reported cases of disease from mosquitoes, ticks, and flea bites has actually doubled over that period of time. And particularly relevant to today's panel, tick-borne diseases accounted over this period of time for over 75% of all reported vector-borne diseases. And on any given year, tick-borne diseases account for approximately 80% of all reported cases of vector-borne diseases. Mosquito-borne disease epidemics are happening more frequently. You're aware of the outbreaks we had actually a couple of years ago of Zika. We've also had outbreaks of chikungunya and dengue, and we frequently have dengue occurring in the US territories. And then finally, the reported data substantially underestimates the actual disease occurrence. And so what I mean by that is that reported cases, this means that when a physician or a healthcare provider reports a case of disease, vector-borne disease in a particular city or county, this person is required to report that case to the city, county health department, eventually to the state health department, and the state health department reports it to CDC on an annual basis. And so, as you might guess, you know, if you were a pediatrician and it's the 15th or 20th case of Lyme disease that you've seen that week, and you're thinking everyone knows that Lyme disease is here, so why should I bother to repeat it? to report it rather. And so, so it doesn't get reported. And so for that reason, the reported cases really substantially underestimates the total burden of illness. And I'll talk about what that burden is in the next few slides. Tick-borne diseases not only are a lot of cases, but they cause significant morbidity and mortality each year. Every year in the United States, there are fatal cases of tick-borne diseases. Most of these are associated with Rocky Mountain spotted fever, with Lyme disease, and specifically with Lyme disease carditis, and then with Poisson virus encephalitis. This slide shows the top 10 notifiable vector-borne diseases in the United States. So these are the ones for which cases are reported. And interestingly, you can see that out of the top 10 notifiable diseases, 6 of these are actually tick-borne diseases, including the top 4. And Lyme disease is by far the highest incident vector-borne disease, and the vector-borne disease for which the most numbers of cases are reported each year in the United States. This slide shows where these cases of vector-borne diseases are typically reported. And what you'll notice along the top panel with Lyme disease, anaplasma, and babesiosis, that they have similar disease distributions across the United States, with a focus on the eastern seaboard, particularly on the Northeast, and then also in the upper Midwest and Minnesota, Wisconsin area, and with cases then on the West Coast, and then a smattering of cases in other parts of the US. So these sort primarily with the black-legged tick, Ixodes scapularis, and in the West Coast with the western black-legged tick, Ixodes pacificus. If you look at the lower panel, these diseases, Rocky Mountain spotted fever, ehrlichiosis, and tularemia, are spread more throughout the central region of the United States, though cases of these occur in other parts of the US as well. But these disease cases are associated with other tick vectors, primarily with the American dog tick, Dermacentor variabilis, particularly for Rocky Mountain spotted fever. And then and then with the Lone Star tick, Amblyomma americanum for ehrlichiosis, and the tularemia is carried primarily by the American dog tick as well. This slide shows the top 10 states for Lyme disease incidence and reported cases in the US just for 2018, and they're ranked according to the first column there, which is 3-year average incidence. And what I'm sure jumps out to you in the slide is that the EPA Region 1 actually is a very important area in terms of the occurrence of Lyme disease, which was already mentioned by previous speakers. But this shows the incidence, and this is showed by incidence. Incidence is the number of cases per 100,000, and so it's not the total number of cases, but the really the rate of cases. And so Maine, while the actual number there is a good bit lower than the number of cases in Pennsylvania, for example, when you weight that by population, the incidence is actually a lot higher in Maine. And Maine, in fact, is the highest incident state for Lyme disease, and it's been that way for several years. This slide is showing kind of a broader map of what we consider the high incident states for Lyme disease, which are shaded in gray. And on top of the shading, you see where the disease cases were reported in 2018. And I just want to make a comment from this slide because I'm sure that many of you have already looked at Massachusetts and are kind of amazed at the absence of blue dots. And all that means is it doesn't mean that Lyme disease doesn't occur there. There are a lot of Lyme disease cases that occur in Massachusetts. And I think our other speakers may talk more about that. But what happened, Massachusetts was so overwhelmed with the numbers of cases that were occurring there that the state, they had a change in their surveillance methods. And so those cases are not actually reported to CDC per the guidance of the CSTE, the Council of State and Territorial Epidemiologists, who actually determine the case definitions and the legal basis for reporting. So those cases are not reported to us at CDC, but they are in fact tracked at Massachusetts at the Department of Health, and they've chosen to spend their resources more toward prevention just simply than counting cases. So but it does sort of point out some of the complications with the disease like Lyme disease, where so many cases occur that reporting itself is an extreme burden to local and state health officials. I spoke earlier about reporting and about underreporting, and I think of reporting somewhat the way you might think of a rain gauge. A rain gauge tells you how much rain occurs, but it's not actually counting every drop of rain. And that's the way that surveillance works. It's telling us about, using a particular definition, how much Lyme disease cases are reported, but it's not counting all the cases, and it was never designed to do that. So what we do to try to figure out more about the burden of illness is we've conducted a number of studies to determine how many cases of Lyme disease, for example, each year actually occur. And this slide shows you some of the studies that have been done and what those estimates have shown you. And if you look down at the bottom line, most recently we published both a lab survey and also analysis of insurance claims data. And what those publications showed us, together with underreporting studies, is that the actual number of Lyme disease cases per year in the US is much closer to 350,000 than just the 30 to 40,000 cases that are reported to us. So I don't need to convince this audience, I'm sure, but all this just really validates the fact that Lyme disease is an incredibly high incident disease, and it causes a huge amount of morbidity and some significant mortality each year as well. To say a little bit about the viral tick-borne diseases, because they're very important as well, even though there's not as many of those that are reported each year. This slide shows 4 of those viral tick-borne illnesses. Only one of those, Powassan, is reportable. The other diseases are not reportable, so that the numbers of cases that we have, particularly for Colorado tick fever, is not it's not a good number, but it's just the ones that we come to find out about that are told to us. But these are not reportable illnesses. We usually hear about these. We usually hear about Heartland and Bourbon cases in particular, because we do confirmatory diagnostics for both of those. But that's sort of what we see with those diseases. And Powassan can be a very highly fatal illness as well. Is transmitted by the same tick, the black-legged tick, that carries Lyme disease, anaplasmosis, and babesiosis. So it's very important in the same regions there, particularly in your region in the Northeast. So moving on to trends, this is what we've seen over the last numbers of years since 2004 in terms of reportable cases. And what I will say is that if you look at the later years, 2017 and particularly 2018, we see these case numbers sort of flattening. And again, those are surveillance artifacts, because in states like Massachusetts, in Rhode Island, and also in New York, a lot of underreporting occurs there, because particularly in New York, in a number of counties, they've gone to case estimates there, so that those cases aren't reported to us at CDC. So I assure you these trends are going up, and tick-borne diseases are getting worse every year. They're expanding both in numbers and in distribution, which is what I'll show on this next slide. This is just a snapshot of 2 years, 2001 and 2017. And in these 2 slides, you get a feel for how Lyme disease cases have increased in distribution over the last 17 or 18 years, basically over the last 20 years. And some of the trends that you see is an expansion, a huge expansion eastward or westward rather from the East Coast. So you've seen it expand across the state of New York, fully across the state of Pennsylvania. You've also seen cases expand northward through New Hampshire and Vermont and into Maine, up the coastal regions in Maine. And again, you can see those trends in Massachusetts and also New York. You see those white areas, and those again are just areas where healthcare providers and health departments have been so overwhelmed by the numbers of cases that they just can't even get them all reported to us at CDC. So I'll talk a little bit about the trends, the drivers that are behind these trends a little bit later, but this is what we're seeing in terms of geographic distribution of cases. And that geographic distribution of cases tracks very well with the geographic expansion of the main tick vector for Lyme disease, which is Ixodes scapularis, which I mentioned before, the black-legged tick. And again, this is just two snapshots. One from a paper that was published in 1996, and a second from a paper that we published in 2015 that looks at the 1996 data and then compares it to the 2015 data. And basically what jumps out at you in this is the areas where the black-legged ticks are now established, which are the areas in red. You can see that these have expanded very significantly over that period of time. And this is from our website. We actually have a program now for national tick surveillance. And this shows a nationwide map of the situation that we have right now in 2020. And what you also see is the California, West Coast region where we have Ixodes pacificus. But this is data now that we provide funding to the state health departments to collect so that we can continue to map these distributions as this expansion continues over the next numbers of years. Also, in terms of trends, I want to say a little bit about what we're seeing with new tick-borne pathogens. And really, if you look on this slide, and we have this data going back to the turn of the century, but basically in the early years, we found maybe one new tick-borne pathogen per decade or so. But then when we got into the 2000s, we've seen this explosion of new tick-borne pathogens. And a lot of this just tracks with the fact that more and more people today are being exposed to the bites of ticks. And these pathogens probably have been out there for many years. And but now with expanding deer populations and expanding opportunities for people to be exposed to the bites of infected ticks, we have more and more cases, and we're seeing more pathogens. And then, of course, you add to this the capacity we now have for detecting some of these pathogens that we didn't know existed previously. So improved detection, improved diagnostics also figures into what we're seeing. But we're seeing more and more pathogens. I could also add to this chart bourbon virus, which we now know is carried by the Lone Star tick, Amblyomma americanum. So finally, just to say a little bit about the causes and drivers behind these trends that we're seeing. Basically, if you look back to the 1800s, as you see in this cartoon in the middle of the slide, much of where you live in New England was farmed. And though over the last 50 to 100, 200 years, we've seen reforestation that's occurred. And together with the reforestation, we've seen a rapid expansion in deer populations. We've also seen a rapid expansion in suburban sprawl, so that you have more and more people living in close proximity to the deer that carry these ticks and to the small rodents that are the reservoirs for many of the disease pathogens. And so you have a greater and greater opportunity for people to be exposed to the bites of infected ticks. And then also, I didn't mention it, and maybe it's a talk for another whole occasion, but a changing climate has had a significant impact on the expansion of tick populations as they move north, because they can now survive over colder winters, over milder winters that are occurring, and there's a longer growing season, and all this contributes to ticks coming out earlier, people being exposed to the bites of ticks, and a longer transmission season. And we've published all of that, and I can give you the publications if you're interested in seeing this. But sort of winding down, currently there are no vaccines for any of the tick-borne diseases in the United States. So efforts focus entirely on preventing exposure to the bites of infected ticks. And there are a number of ways that you can do this, and the other speakers are going to talk more about that. But, you know, we just emphasize wearing repellents, doing tick checks, throwing your clothes in the dryer on high heat for 10 minutes after you've been out in the area where ticks might be, doing showers, and of course, if you removing ticks promptly, and then if you get a fever or illness during the summertime when you might have been exposed to a tick, go to see your healthcare provider. And all of these resources are available at our web page. Very simply, www.cdc.gov/ticks will get you there to all of these resources. So in conclusion and summary, tick-borne diseases are very important public health concern. They are increasing in numbers and distribution, and also in the numbers of new pathogens that we're seeing. The drivers for these are related to increasing exposure to the bites of infected ticks. This is largely influenced by increasing deer populations and other changes in the natural and in the built environment. And then in the absence of vaccines in the US, primary prevention focuses on reducing exposure to ticks and quickly removing any ticks that are found on people or on their clothing. So I will stop there and turn it over to the next speaker. Thank you.
Thank you, Dr. Beard. Our next speaker is Dr. Kirby Stafford with the Connecticut Ag Experiment Station. And Dr. Stafford will concentrate on or focus on tick basics and tick management and control.
Good afternoon, everyone. Today I am going to cover some tick basics and how to manage and control ticks. And I'd like to start the presentation with a quote from Dr. Dan Sonnenshine's book, Biology of Ticks. Few agricultural or health problems confronting human societies have proved as intractable as the control of ticks and the many diseases that they transmit. And that's because ticks are found almost everywhere in wooded and successional habitats in relatively high numbers. And remember, ticks don't fly. You know, people must enter or live in tick habitat to become exposed. And many homes are built in forested tick and host habitat, creating a large degree of residential exposure. People also visit and camp in parks and state and national forests where ticks are found. So basically the environment, the presence and abundance of the host of the tick, as alluded previously, and the geographic distribution of the various tick species will really determine your potential risk of exposure to ticks and tick-borne diseases. Now, while the infection prevalence may be somewhat predictive of the transmission risk for a specific tick-borne disease, it's really the abundance of ticks and the number of tick bites that people receive that increases your chance of encountering at least one infected tick. And so, consequently, your actual risk of exposure is dependent upon human behavior, how much you're using personal protection measures, and conducting those tick checks. So here in the Northeast, the exposure appears to be largely residential based on the results of active, or I should say passive, tick surveillance in Connecticut and in Monmouth County, New Jersey, where up to 74% of the ticks submitted to the experiment station or other labs were acquired outdoors, right at home. But 21% were acquired in activities away from home, or 18% in parks. So basically, even though exposure is largely residential, about 20% of the tick exposure is probably occurring in recreational activities in our various parks. Now, there's about 16 to 17 species of ticks in the northeastern United States, if you include New York and New Jersey, but there's 3 species that commonly bite humans. The black-legged tick, Ixodes scapularis, actually accounts for 80 to 83% or so of the submissions to the Connecticut Agricultural Experiment Station. The American dog tick, Dermacentor variabilis, accounts for 13 to 17% of the submissions to the Agricultural Experiment Station for identification. And then about 3% of the submissions in the past few years have been of the Lone Star tick, Amblyomma americanum, and that's actually up to over 4% here this year, and I'll be talking a little bit more about that tick in a moment. And we also get through 2 or 3 woodchuck ticks, Ixodes cookii, submitted as well. And also, in addition, occasionally we'll see a number of exotic tick species being submitted from those who have been traveling overseas. And we also now have a new exotic Asian longhorn tick, Haemaphysalis longicornis, here in the United States. It's been detected in about 12 eastern states, and it's quite abundant in parts of New Jersey and New York. We've only picked up a few specimens here in Connecticut so far, but it's something we're keeping our eye on. Now, the one thing about all these ticks is they're 3 host ticks, exemplified by the life cycle here of Ixodes scapularis. The larval ticks hatch from the eggs and then feed predominantly on rodent and bird hosts, as you see in number 1 on the left, but they will feed on any animal. After the larval ticks engorge, in August is the peak month then. They drop off, molt to nymphs, and the nymphs actually pass through the winter, emerging the next summer. And again, feeding mainly on rodents and birds, but nymphs will feed on any animal, and they're actually responsible for the majority of Lyme disease cases. Once the nymphs feed, they drop off, molt to the adult stage, and the majority of female ticks actually feed on white-tailed deer. They do not feed on the rodents and birds, they feed on medium to large animal hosts, and each female tick, when she drops off, will lay about 2,000 eggs, and this whole life cycle takes about 2 years. And as I mentioned, one of the things that we're keeping an eye on is the Lone Star tick and its range expansion here into the Northeast. This tick moved into Long Island, New York in the early 1990s. This tick is responsible for about 90% of the tick bites in the southeastern United States and is becoming an increasing concern here in the Northeast. It has now become established in some new areas along the southern New England coast, and we're detecting more and more of these ticks through both our passive and active tick surveillance activities. We've actually done some studies. We've shown that this tick can successfully overwinter in Connecticut, and also to some extent even up in coastal Maine. So under our current environmental conditions with warmer winters, our moderate maritime climate, all of this favors the establishment and expansion of the Lone Star tick along the New England coast. So they're going to be an increasing problem here in the Northeast going forward. So what are we going to do about these ticks? Well, there's a number of approaches to integrated tick management, and I'm going to kind of give a quick overview of these here today. Obviously, as mentioned earlier by Dr. Beard, your personal protection measures is your first line of defense. There are landscape and vegetative modifications that can be done to kind of reduce the suitability of the habitat for the ticks. The mainstay for tick control has been chemical control, primarily synthetic acaricides, but there's increasing interest in the use of so-called botanicals, which are basically essential oil-based products. There's been work done on biological control, that is biopesticides, as well as host reduction or exclusion, host-targeted acaricides, and there's also been research done on host-targeted Lyme vaccines for the mice. And this is not available yet, so I'm not really going to talk about it here today. Of course, as previously mentioned, personal protection measures is your frontline defense. Basically wearing appropriate clothing, pants tucked in socks. Most of the ticks are acquired on the lower extremities. And then use a repellent, either DEET or picaridin or oil of lemon eucalyptus on clothes or skin. You also have an option of using a permethrin clothing only repellent available as a spray, or you can also get permethrin pre-treated clothing, which provides a high level of protection against tick bite. And information for sources for tick repellent use and effectiveness are shown here. The EPA has a website for helping to select insect repellents, and the National Pesticide Information Center also has information on how to appropriately select repellent for your family. When you, as I mentioned, get back, do those tick checks and promptly remove them. I prefer fine tip forceps for removing a tick because the nymphal stage of the black-legged tick is extremely small. And as you can see in the upper right-hand corner, just a small straight pen with an engorged nip for size comparison. So remember that repellent when you're hitting the trails, and do that tick bite prevention. So this headline indicates experts are concerned that COVID-19 pandemic could lead to a bad tick season. Medical experts fear that many people who've been stuck inside due to the COVID-19 pandemic may let their guard down once they're able to venture outdoors and not pay attention to tick bite prevention. And as the sign says in one locality, tick alert. Ticks carrying Lyme disease can be found in this area. Examine skin, clothes, and pets thoroughly. So when you're walking outdoors, you know, keep those pandemic precautions in mind, but do those tick bite prevention measures. Keep to broader trails if possible to reduce your likelihood of encountering vegetation harboring ticks, and make sure you're using repellents and other precautions, particularly if you're on those more wooded, brushy, riskier trails shown on the right. Landscape management is another approach. Most ticks require high humidity and cover, that is ground or vegetative cover, for survival. Ticks don't jump, they don't fly, and are not dropping from the trees. Most are found in the leaf litter, ground cover, or lower vegetation in the woodland or ecotone areas. Actually, most of our ticks spend 90 to 95% of their life at or just below the ground cover in the duff layer, either digesting a blood meal, molting to the next stage, in diapause, or actually seeking a host. So those ground conditions are critical to their survival. So when you think about schools and parks and campgrounds, you know, parts of those areas may already be landscaped into what I call a tick safe zone, with lawns and mowed fields and playgrounds and buildings, and also depending on the amount of tree cover. So often it's that landscaping at the edge that's really important, or walking through those trails that I mentioned earlier. And these numbers apply mainly to residential areas, but some earlier studies found that 67% of black-legged ticks are in the woods, 22% can be found in that ecotone area, and only 2% of the population is out in the lawn or more open areas. But nevertheless, of that 2%, 82% on the lawn are within that 3 meters of the edge with the stone walls and forested and ecotone areas. But nevertheless, about 18% of the ticks can be found beyond those 3 meters. So that's your high risk zone, but bear in mind that's not your only risk zone. And of course, higher tick counts have been associated with exotic invasive forest understory as opposed to native forest understory or open understory forest. So control of invasive plants is an important component for the management of ticks. These dense stands of Japanese barberry or invasive honeysuckle provides an ideal microclimate for the ticks and great host habitat. So basically, the reduction in long-term management of barberry has been shown to significantly reduce the abundance of infected ticks. And similarly, the removal of honeysuckle has decreased deer activity in the numbers of infected ticks as well. So that could be an important component of your management strategy in not only residential areas, but parks as well. Spraying continues to be the mainstay for controlling ticks, particularly on residential properties, and these may consist primarily of synthetic acaricides like your carbamates, your pyrethroids, neonicotinoids, or animal-only products. Microbial biopesticide is another option with Metarhizium anopheles. This is an entomopathogenic fungus under the brand name Met52, and you can see a female black-legged tick in the center there that has been killed by this fungus. And as I mentioned earlier, botanical products based on plant extracts or essential oils has been a focus of a lot of recent interest, and these are mainly materials derived from the EPA's 25B list of minimum risk pesticides. But how well do they work? Well, the synthetic pyrethroid insecticides or acaricides are actually quite effective, as you can see in this chart here, where you can get almost up to 100% control with these compounds lasting for a period of weeks. The carbamate carbaryl, or commonly known as 7, can also provide high levels of control for weeks to even months. The biopesticide with the fungus Metarhizium anopheles can also provide moderate to high levels of control for a period of weeks. There is not yet a lot of published information on these 25B based essential oil products, but one product, IC2, which was a rosemary oil based product, it's no longer available, but in one published study, they found relatively high control with this. So there's a lot of focus and research right now on evaluating a number of these products, but I will say it seems to be somewhat of a mixed bag, with some working relatively well and others not working at all. So we'll just have to keep our eyes open on that subject. Host-targeted tick control has been a focus of a lot of research, targeting either the white-footed mouse, which is the reservoir and main host for immature stages, as well as white-tailed deer, which is the main host for the adult stage of not only the Lone Star tick, but the black-legged tick as well. The one thing to bear in mind when you're thinking about host-targeted measures is the home range of these animals. So a white-footed mouse home range runs from about 1/10 of an acre to 4.5 acres, with an average of 0.7 acres. So your intervention has to be on that kind of a scale. By contrast, deer have a urban home range ranging from tens to 100 of acres. One study in Old Lyme, Connecticut, the average home range was 142 acres with a core of around 20 acres. So there are 2 commercial products available today that target the white-footed mouse and to a lesser extent the chipmunks. These are the permethrin-treated cotton balls in the tick tubes, and the fipronil bait boxes, which consist of 2 non-toxic food blocks and a wick with fipronil. Fipronil is the active ingredient in the product Frontline for flea and tick control on dogs and cats. Bear in mind, chipmunks do not use these tubes, and these products, as getting back to that home range, are placed every 30 to 60 feet around the perimeter of the treated property. So they're probably not going to be practical or cost effective at the scale for most parks or recreational areas. And similarly, they're not going to be applicable for the control of Lone Star ticks, as the immature stages of this tick don't readily use rodent hosts. A lot of work is focused on white-tailed deer, and there's been 3 main approaches: either excluding the deer, reducing the deer, or treating the deer. Fencing the deer out on a scale of acres can reduce the tick population substantially, 100% for larvae, up to 84% of the nymphs and 74% of the adults. The deer aren't coming in and dropping off female ticks, you're gonna have fewer ticks. There's been a number of studies, particularly on geographically isolated islands or tracts, with reducing deer populations, and it appears that if you can get deer down to around 13 deer per square mile or lower, you can reduce the tick numbers sufficiently to even potentially impact the number of cases of Lyme disease. And then lastly, treating deer is another option, this through a 4-poster device. This was developed by the USDA Agricultural Research Service. It holds about up to 200 pounds of corn. When the deer come in to feed at a little trough on either end, they brush up against the paint rollers that are treated with permethrin. And there's been a lot of studies with this. And if you're more interested in this, I did publish a review of this approach in the Journal of Integrated Pest Management. This is open access, and readily available to anyone who's interested. But no one single tool has seemed to be the solution to the problem. So there's been an increasing interest in an integrated approach, targeting rodent hosts, spraying, and so forth. So I don't really have time to go into that here today, but I would refer you to 2 papers in the Journal of Integrated Pest Management that are open access. One is the one that I published back in 2017 on integrated pest management and controlling ticks and tick-associated diseases, and another paper by Alexis White and Holly Gaff, which is a review of the application of these tick control technologies for black-legged Lone Star and American dog ticks. Nevertheless, I will highlight one example of tick IPM in a recreational area. This was a study done to control Lone Star ticks at Land Between the Lakes, Tennessee, where they looked at 3 different combination treatments, either acaricide and vegetative management, acaricide applications and host management, which was fencing, and then acaricide applications, vegetative management, and host management all together. And they produced a 94, 89, 96% mean control of all life stages respectively. So it is possible. They also established an economic threshold for sampling ticks on carbon dioxide traps based on an attack rate of less than 1 tick per human visitor per day. Anything above that, people stop coming to the park. So it has been a difficult issue in terms of tick management, and there's been a number of barriers to effective tick management that are highlighted in this paper that was recently published in the Journal of Medical Entomology. It's in the public domain, open access. And some of the issues that were identified, and I don't have time to go into a lot of detail on these, include things like the skepticism and public distrust of chemical pesticides and repellents, the social acceptability, or I should say lack thereof, of deer management, how much people are willing to pay for effective tick control measures, as well as the lack of funding or resources for large-scale neighborhood or community area-wide studies. And there's also concerns about pesticide resistance, as well as things like pollinator health. So effectiveness, the scale of the tick management tools, the cost of those tools, and whether they're actually implemented are key components for tick management strategies. So where do we go from here? Ticks are widespread and difficult to control. The growing number and spread of ticks and tick-borne diseases pose an increasing risk in the U.S. There are many tools available for killing ticks, but the impact on disease remains unclear or unproven. There are multiple challenges or barriers to effective tick bite prevention or control. Few of the methods are available or utilized by homeowners or perhaps applicable to parks. We need more tools that are safe, scalable to larger areas, and that are cost effective. And with that, I thank you for your attention. My contact information is given here, and I will gladly turn it over to our next speaker.
Thank you, Dr. Stafford. Sort of sobering. Our final speaker is Dr. Steve Rich with the University of Massachusetts. Dr. Rich runs the tick testing lab and is a professor of microbiology at University of Massachusetts Amherst. Dr. Rich.
Thanks, Rob. Well, it's a real pleasure and an honor for me to be part of this lineup. So, and I thank you for that invitation. What I'm going to try to do today, in just a brief amount of time left here, is try to tie together some themes that were touched on in the first 2 talks and present a view for how we can go about assessing tick-borne disease risks. And my assertion to start off with is that every tick-borne disease can be thought of as a story. And so like every good story, it will have a beginning, a middle, and an end. And very often we focus, when we're thinking about tick-borne diseases, the beginning of the story and the end of the story. And I'd like to talk about what can be done in the middle part of that story. So quite specifically, in the beginning of the story, we can be concentrating on how to control the number of ticks that are biting people, how to reduce the number of ticks that are biting us as individuals. And at the end of the story, we can be thinking about the preponderance of disease, the frequency of disease in people that have failed to keep ticks off themselves. What I'd like to talk about in the middle is actual things that can be done to assess the risk where a tick has bitten a person, and understanding that not all tick bites are equivalent, we can assess the risk of that tick bite and make some decisions based on that assessment. So I'm hoping that this will be of interest to your listeners who may be encountering ticks themselves and or their constituents are encountering ticks. So that's where I'm going to take this. So when we think about public health exposures in the parlance of public health theory, we can think about risk and hazard as being two aspects of the threat, and exposure is the piece that mitigates and leads to the threat itself. So let me give some concrete examples here. We can think about sharks as a potential threat to public health, a shark attacking a human. And so the hazard there is something that's a potential harm to us. And this will ring true for a lot of people that think about environmental risks, because although this is a biological and a vector-borne problems, in many ways, it's more analogous to environmental problem than other infectious disease because it's a zoonosis and the threats themselves are in the wild. So the hazards here are the things that are present that pose a potential threat. And then, of course, risk is that other end of the story, which is the likelihood of the hazard actually causing harm. So this is the endpoint. So how many people are suffering from, in this case, shark attack? And so if we take that analogy and we think about the mitigating piece, the exposure piece, and transfer that over to what our topic is today, which is tick-borne diseases, then we can say, yes, there are hazards which we can go out and we can measure and we can quantify the number of ticks present in the wild. That's one interesting aspect of the threat evaluation. And then, of course, there's the risk factor. So we heard a lot about the CDC and the state data from state public health agencies about who's actually getting sick. So this is images showing someone suffering from the telltale signs of Lyme disease in this case. But very often it's that exposure piece, that mitigating piece that is of greatest concern to us that are in the middle of that story, that aren't currently suffering from severe disease and or may have been bitten by a tick. So that's what I want to focus on just in the few minutes we have left here. So we think that it's important to measure exposure by the three characteristics that determine the threat level of a particular tick exposure or tick encounter. And those are the tick species, the duration of feeding, and the infection status of ticks. So as we've already heard, there's more than a dozen different ticks here in New England. There are more ticks still from across the country that bite people. And so not all tick species are created equal. So too, the duration of feeding of ticks is highly relevant because, without going into a lot of detail about this, the ticks don't function just as syringes that suck blood up or spit saliva in. They actually undergo physiological changes as they feed, and so too the pathogens that are inside those ticks will change over time, and that takes time. And if the ticks haven't fed long enough, the contents of that tick may not get out of that tick into an individual. So in some cases, the tick could be the right species, but if it doesn't feed long enough and or it's not infected, then it poses less risk. And we would say the exposure is lower for that one. And then the last factor is, of course, the infection status. So we've already heard from the previous two speakers that there's a percentage of ticks that are infected. Maybe 50% of the adult deer ticks in New England are infected, and maybe less, 25 to 30% of the nymphal ticks. For other pathogens, those numbers are much smaller. And so the infection status is very important for evaluating a tick bite. And the upshot here is that what people should understand is that not every single tick bite poses the same threat of a long-term health problem or a long-term bad outcome from that tick exposure. So we do this. We make these evaluations from people that send us the ticks that have bitten them. And I like very much the analogy that Dr. Baird used of measuring the rainfall, not the drops of rain. And point of fact, what we're doing is measuring the drops of rain. So the drops of rain are ostensibly sent to us as people encounter them. That's the tick bite. And then we make these evaluations of the species of tick, the duration of the tick's feeding, because we can tell how long the tick has been attached based on changes in size and shape, and in some cases coloration of the tick. And then of course we can look at the status of infection, because we can screen them for a number of different pathogens which go beyond, although I know we're focusing today on black-legged ticks, there are other ticks that feed on people, and there are other pathogens associated. So in short, I think what we've heard about for how we go about looking at the threats, in this first two columns, you see that something we've done for a long time is field sampling of ticks. So this is the active surveillance. This is going out, like Dr. Stafford was describing, and collecting ticks in the wild, bringing them back and testing them for infection. But I would argue that that's not necessarily a direct measure of the rate at which people encounter ticks. So for example, if you go to a site, a park or a forest that perhaps isn't visited by many people, there could be lots of ticks there and lots of infection. But if a lot of people don't go there, it's not really reflecting on the threat overall. You can get a direct measure of infection rate, but you don't get any information about duration of feeding. So too with human case reports, when you look at the number of people that are reporting illnesses, you get an indirect measure of the infection rate, because presumably all those people that are sick were bitten by a tick, but you don't know how many ticks it took before they got an infectious bite, or perhaps even how many infectious bites it took before they got infected. And so in the third column, you see what I think is really another way to look at this, which is to do surveillance of human biting ticks. And again, Dr. Stafford's group in Connecticut does this. We do this on a national scale, and it gives a direct measure of these different levels of the exposure, the tick encounter rate, the infection rate, and the duration of feeding. So the first two measure hazard and risk, and we're trying to get some quantification of exposure. So let me just give you a concrete example of how this manifests, the differences between looking at the risk or hazard. So this is the distribution of the hazard associated with Lyme disease, which is the black-legged tick, the distribution of black-legged ticks. And if we superimpose by these dots, and this looks very similar to what Dr. Baird showed earlier, if you look at the distribution of Lyme disease cases, you can see that they're overlapping, but they're not completely concordant. So there's a lot of Lyme disease in the Northeast and upper Midwest, but there's not a lot of Lyme disease in the Southeast where the ticks are present. And so something's going on here, and I think that's just one little example of why it's fundamentally important to look at those exposure factors. And we think there's a bunch of different things going on in the Southeast that aren't going on in the Northeast, or vice versa. So what we do is we test ticks for the pathogens that are most commonly associated with the tick species. So there's a list of this pathogens most commonly associated with black-legged ticks. So this includes Borrelia burgdorferi, the causative agent of Lyme disease, a relatively new Borrelia called Borrelia miyamotoi, which causes a different type of disease. It's a relapsing fever-like illness, and then Borrelia mayoni, which is a relatively narrowly geographic range, Anaplasma phagocytophilum, Babesia microti, and then something called Ehrlichia miris-like agent. We also test for a Powassan virus. So we were actually the first lab to start providing testing for for that pathogen. It involves a special set of tricks in order to be able to test for viral pathogens, because they're RNA-based and the RNAs tend to be less stable. But we don't just test deer ticks, we also test non-deer ticks, because what we know is that there's a non-zero threat associated with those as well. So while the incidence of things like Lyme disease in non-black-legged ticks is probably next to zero, there are pathogens associated with those non-ticks, so we test those as well. And so here's I'm going to show you a couple of summaries of the data that we find nationally. So like Dr. Stafford reports, we also see the primary canonical human biting ticks, the black-legged tick, the American dog tick, and the Lone Star tick shown there at the top. We also see a smattering of Western black-legged ticks. That's the Oregon, Washington, and California human biting tick associated with Lyme disease transmission, and then Pacific Coast tick and Rocky Mountain spotted tick. But you can see that we see a number of rare tick occurrences as well. So some relatively rare things like woodchuck tick that Dr. Stafford mentioned, generally a nest parasite, so don't come out of the nest very much. But you can see in this case, we've seen 157 instances of that. Or things like the squirrel tick or the Asian longhorn ticks were associated with, in some cases, being on humans. So again, the value of not just looking at what ticks are in the wild, but also actually quantifying the ticks that are biting humans is fundamentally important, because each of these could pose unique exposures to unique sets of pathogens, which typically aren't thought of as being human pathogens. So in the end, there's a public health benefit to this, to making these assessments, and we refer to this as a passive surveillance. And again, it came in the previous slides as well. And we've created what we think is a passive surveillance network. And if passive surveillance is a term that's not familiar to you, passive surveillance, as opposed to active surveillance, is the instance where the samples actually come to you. So in this case, the ticks are sent to us. So in that regard, our collection is a passive approach, as opposed to going out and counting the number of people that are ill or the number of ticks that are infected. And they both, active and passive surveillance, measure the same underlying distributions in a sense, and yet there's cost effectiveness to passive surveillance and sustainability to it, obviously. And in most cases, we think that the passive surveillance will reflect active surveillance in a number of instances. It can also reveal some fundamentally important details of those exposures that you don't get when you just look at infected people or infected ticks. Now, where members of this panel might have some disagreement is whether that individual information about positive and negatives has any individual benefit. So the CDC has a very strong position in saying that they are concerned, and we share that concern, about whether people should interpret a tick test as a diagnosis. And I'll be clear that it is our position that a tick test should not be thought of as replacing or in any way representing a diagnosis of human disease. It is, however, a very good tool for evaluating the exposure risk of an individual tick. And so, notwithstanding the fact that it's possible that someone can have a positive tick test and not get infected because of the things that we talked about, the other exposure factors we talked about, or vice versa, it's possible that someone could have a tick pulled off them that was negative for pathogens, and yet they still could get the pathogen because they got bit by a second tick. Notwithstanding those caveats, we think there's a great deal of value to being able to assess individual tick bites and then make decisions, in some cases, about what to do next. So this is a little diagram that came from a paper by Alan Steer from back in 2004. Alan Steer, for those of you that don't know, was the doc that originally described the syndrome of Lyme disease here in the United States in Old Lyme, Connecticut back in the 1970s. And so he wrote this paper in 2004 where he describes an algorithm for what can be done, the courses of action to be followed after certain summer events. So notice here, there's a tick bite and then separate off for skin rash and summer flu. So again, you can think of these as parts of a tick-borne story. So here's a tick bite. Here's what happens after the tick bite. You develop a skin rash and then eventually perhaps summer flu-like symptoms. And what Steer pointed out is that in most cases, human testing, so testing the human blood in the first couple of weeks after these occurrences is not of much value, because the tests are based on detection of antibodies which are responsive to those pathogens. And so in the first couple of weeks after one of these events has taken place, particularly the tick bite, it's not going to be particularly effective. And so he's pointed out that you don't need to really test in order to know, to reach a clinical conclusion that there may be a Lyme disease risk. And so there can be a treatment, for example, an oral therapy with just 2 doses, the prophylactic therapy, or a longer course if there's indications of disease. Our contention is that there must be some place for evaluating the exposure risk, that there must be some non-zero value associated with assessing an individual tick bite, particularly in cases where antibiotics are otherwise contraindicated, where the kid is too small, a young child, or so there's an allergy or whatever reason they're unable to treat, or in some cases where people are insisted upon treatment and yet the indications are that, you know, I'll say straight up that we test ticks that sometimes we are sent specimens sometimes that turn out not to be ticks at all. We get sesame seeds, we get scabs, and so there again, we're also able to inform that individual and hopefully can be brought into the conversation with the physician that what appeared to be an exposure risk was not an exposure risk. And again, underscoring that it's not a diagnosis, but that there is some value to having that information about that exposure, we think is just a logical conclusion. And so those first three boxes at the top on the left are associated with Lyme disease risk. For those other non-Lyme disease risks, we think that tick report or tick testing in various kinds can be in some ways more valuable, because if you can imagine a scenario where someone has a negative serology or a negative blood test for Lyme disease, and yet they're still suffering from some illness, it could be associated with another pathogen. And so we have a growing number of anecdotes where we have tested ticks that were infected with particular pathogens, and then subsequently human testing manifested that that person had been infected with that. And we know in at least one of those cases where we reported it out as a published case report, that the tick report led to that blood test that eventually led to the appropriate diagnosis. So in conclusion, I just want to say that we provide this tick testing because we think it provides a valuable passive surveillance information, and also that we think it's valuable to individuals. But I also wanted to point out that we've learned over time a lot about people's tick encounters, and so we, through our FAQs and through an online chat tool, as you can see down here in the lower right-hand corner, we, not just as subscribers of the service, but because it is a fee for service, I should mention, but we also provide to just individuals that have been bitten by a tick, we can provide information about the common things that they might think about after that tick bite. And we think this is in many ways much more powerful than a brochure or a web page statically, because we can answer these things in real time through the tools that we've developed, online tools for stats, for chats, etc. And then the last thing is that we make these data that we collect publicly available through our statistics page. So if you go to our webpage, www.tickreport.com/stats, you can go to this webpage, tickreport.com, and click on the statistics icon. You can see the data for the 80,000 plus human biting ticks that we've tested since 2006. And so even if you're not going to test ticks in your zip code, you can search and see what ticks have been sent to us in this 14-year period from that zip code and see what their infection status is. And I remind you that we stay on, so the chat line that we have available, we answer things, the average response time is 3 minutes, and that means from 9 a.m. to 9 p.m., we answer almost instantaneously. If you send us something at 3 a.m. in the morning, we're probably not going to get answered till the next morning via email, but we do get back to everything. And so I'll just conclude by saying this is, Tick Report, we're here at the university. It's a team effort. We have 10 full-time technicians, a smattering of undergraduate students and graduate students that are involved with the project, and we're very proud of what it provides as a public service, and we think it has an important place for making these risk assessments. So with that, I'll say thank you and turn it back over to Rob.
Thank you, Dr. Rich. Thank you to all the panelists. Doctors Ben Beard, Kirby Stafford, and Steve Rich. Now is time to pose questions using the chat space to the right of your screen, indicated with the red arrow. As a reminder, you can download today's presentation by pressing the paper icon, and you'll be able to get all the links and resources by doing that. For the Q&A session, for the question and answer session, which will be about 15 minutes, we have invited representatives from each of the 6 New England states to participate. While, of course, most questions will deal with the presentations themselves, we thought there might be some questions that deal with state-specific pesticide issues. EPA works closely with states on implementing pesticide laws, and the states have primary responsibility for licensing and certifying applicators and for enforcing pesticide use laws in their states. The state lead agencies, all 6 states are represented, so if you're from any of the New England states, feel free to ask about state-specific issues. This slide just indicates who will be represented from each of the state lead agencies. A couple of announcements before we begin the question and answer session. First of all, we have some resources, and then finally upcoming events. So in terms of the resource, each of the state lead agencies has provided their website, and so information on their state pesticide programs are located on these websites. So Connecticut, Maine, Massachusetts on this slide, and New Hampshire, Rhode Island, Vermont on the second slide. Beside that, EPA has a lot of useful resources. Listed here is the EPA Region 1 website, followed by the Office of Pesticide Programs website. The Office of Pesticide Program website has an incredible amount of information on everything from registering pesticide products to how to control bed bugs to the third link, Pesticide Program Tick Resources, and the last link there, COVID-19 resources. Assistant Administrator Dunn mentioned coronavirus. EPA does regulate antimicrobial products, and there's an excellent website, which again is listed on the bottom, coronavirus. So in terms of upcoming events, we have the companion webinar, which is scheduled for July 16th. So with that, it looks like we have a large number of questions already. I'm afraid we may not be able to get to every question, but we'll cover as many as we can. So our first question looks like do insect repellents provide adequate protection for campers and people in recreational areas? I guess Dr. Stafford?
Repellents can be actually quite effective if you're using deep picaridin or oil of lemon eucalyptus, which can be used on clothes or skin. You do, for ticks, do want to use somewhat higher concentrations in the 20 to 30% range, as opposed to say a 5% formulation to provide a decent amount of protection. The permethrin, which is an insecticide for only use on clothes, provides extremely high levels of protection, and that might be a good option for, yard work or activities where you're willing to wear those long pants and tuck them in the socks and so forth, and you can buy pre-treated clothing as well. I will mention that in the many years being in the field doing tick research, I've used the permethrin and I've had very few tick bites, and actually I have not gotten Lyme disease yet.
Well, thank you. Here's a question. Building and grounds manager from Maine Organic Farmers and Gardeners Association. I've been diagnosed with Lyme disease twice, and once 20 years ago on Eastern Long Island, and several years ago here in Maine. Over the years, I've used chickens and ducks as part of my IPM strategies. Are there any studies on these types of techniques? I guess Dr. Stafford again.
Yeah, there has been some interest in guinea fowl and chickens for trying to manage ticks on properties. There's very little evidence that they actually really work. Bear in mind, too, that birds are a major host for the ticks as well. So they can actually be a host. There was a few little studies. There was one study that looked at turkeys and found that they really were not consuming even the adult stage of the tick. And of course, the larvae and nymphs are even smaller. It is possible that they might be conceivably consuming some of the engorged female ticks down in the leaf litter as, let's say, the chickens or guinea fowls are scratching through it, but those studies haven't really been done.
Okay, well, here's a question on the distribution. So maybe Dr. Beard, why does the distribution of various diseases and ticks seem to be concentrated in the upper Midwest and the Northeast?
Yeah, that's a good question, and I think that probably it certainly is related to the distribution of the black-legged tick, Ixodes scapularis. And you know, the other speakers can comment on this, but sort of if you look back at the history of Lyme disease, you know, the first cases were reported really it was first discovered of course, in coastal Connecticut, in Old Lyme, Connecticut, and this was in the early 80s. But it's very interesting that at the same time very near to the same time that it was described there, you know, the cases going back, you know, into the 70s, that it was also reported in Wisconsin. And there was, you know, a famous account, I consider it famous, really interesting. It was a physician hunter who had a tick bite and classic Lyme disease, erythema migrans, fever, and all this. And he successfully treated himself with a round of doxycycline or tetracycline. And but for all intents and purposes, Lyme disease really existed in both of these areas. Going all the way back to the late 60s and 70s. And then, you know, it's been found retrospectively in museum ticks and things like that in New England. And so my view on this is that Lyme disease is not an emerging disease so much as maybe a re-emerging disease, and that you know, it's been around here for a long time. Who knows whether it was brought over with settlers, you know, European settlers, if it was here prior to that, because we also have it, of course, in the West Coast, in California and the northern seaboard in the West Coast. And so sort of the story I told with reforestation and what I've read about this and heard about this is that deer really had been hunted almost to extinction. You know, the numbers were really low, and much of the land was plowed, and it really wasn't a good habitat for deer either. And then over, you know, the 200 years or so, we've had this huge reforestation. And so, you know, if you think about the expansion back from small areas in the Northeast and small areas in the upper Midwest, that has really expanded from there and re-expanded, and in close association with the small rodents, the Microtus, and the white-footed mouse, and then with Ixodes scapularis, the deer, the black-legged tick, and then deer, who are the important reservoirs for the, or hosts for the ticks. So all that's been re-emerging in these areas, and so the distribution that we see right now really focuses on this natural transmission cycle with the tick, the deer, and the rodent, and how this has been re-expanding. And as Dr. Rich mentioned, we also see Ixodes scapularis in the southern U.S. And but you know, when Lyme disease was first described by our colleagues at Harvard, originally the tick vector was called something different. It was Ixodes damani, and then later genetics studies revealed that damani scapularis were actually the same species, but scapularis being the southern and damani being the northern variant of these, and of course then that was the way people saw this. And now we know that they are all one species, and but those ticks in the south behave quite differently. And there's most likely genetics behind this, that those populations of ticks in the south live down in the leaf litter. They typically don't come up higher to quest for people, and many of them bite lizards that are down in the leaf litter, and lizards actually are incompetent reservoirs for the Borrelia that causes Lyme disease. So there's a different ecology and behavior in the south with those Ixodes scapularis populations than the populations that we see in the north. And interestingly, what we're seeing is that population behavior or phenology that we see in the north that has been associated with Lyme disease seems to be expanding southward. So we've seen that line where ice caps are actively biting people. We've seen that line move south. So now we see transmission of Lyme, significant transmission occurring, in the Carolinas and West Virginia and further south each year. So it's an interesting phenomenon, but I hope that sort of answers the question, and one of the other speakers may want to comment further if it doesn't.
Yeah, no, I think that's good and interesting. We have a question here. How would you control ticks on natural reserved near the seaside? I guess Dr. Stafford, natural areas and controlling ticks and sort of unique habitat type things.
Yeah, controlling in natural areas can be quite a challenge. In many cases, there are strict guidelines that prohibit the use of acaricides. Some vegetative management, like getting rid of some of the invasive plants like the barberry, honeysuckle, things like that might be a more acceptable approach. Managing, the deer population could be an important element as well. But generally, chemical control is generally not allowed in natural areas.
Okay, well, here's an interesting question. There's, you mentioned some plants, invasive plants that seem to create habitat for reservoir species, I guess, but are there plants that can be used, either native or non-native, that kind of deter ticks or, are not attractive to ticks?
From a practical standpoint, there's no information that would suggest any plants are repellent in and of themselves in their natural environment. It's just that it's very clear from some studies that tick abundance is much higher in these areas with dense stands of invasive plants as opposed to the native vegetation or forest areas that have grown up so much that there's very little understory at all.
Okay, unfortunate. Are we close to a cure for Lyme disease? I guess Dr. Rich or Dr. Beard?
Well, I can comment on that. It depends on what you mean in terms of cure. There's no vaccine available. There's a vaccine actually that's in phase 2 clinical trials right now, and there's a lot of hope that that will be very useful. Lyme disease, the secret to curing Lyme disease is to diagnose it early and accurately, quickly. The sooner it's diagnosed and sooner treatment begins, the more effectively it can be treated. And there are a lot of patients who are effectively treated and cured of Lyme disease. Part of the problem, there's two problems. So one is that if you live in an area where Lyme disease is endemic, getting cured one summer doesn't mean that you're not going to get it again, the next summer or the year after that. I mean, if every year is a bad year for Lyme disease, if you live in an endemic area, like I would say most of you probably do. So that's one point. The other point really quickly is that if Lyme disease is not treated early, then it can be very difficult to treat. And patients can go on to develop late manifestations of Lyme disease, neurologic manifestations, arthritis. These can be difficult to treat. And in a subset of patients, once they've been treated, they can go on to develop persistent symptoms. And there's a lot of confusion and debate currently over what is the cause of those symptoms, whether it's persistent infection or whether it's autoimmune responses or inflammatory disease or illness. And related to that is a lot of controversy over the safest and most effective way to treat those patients.
Okay, well, thank you. And it's 3 o'clock, but there's 2 more questions listed here that I'd like to ask, and I'll ask them both at the same time. How often are ticks found to be infected with more than one pathogen? And the second question is, what is the cost per tick for tick testing? And I guess maybe since both Massachusetts and I think New Hampshire or Connecticut have tick testing programs, I'll ask both of you to respond to the questions. This is Dr. Stafford and Dr. Rich.
So the tick testing program at the University of Massachusetts is a nationwide program, so it's not supported by the state or federal sources. The cost is, the entry level cost is $50, and there are additional fees for testing for additional pathogens. The answer to the first part of the question is we see roughly 2% of ticks in some endemic areas are infected with more than one pathogen. We've seen ticks that are infected with up to 4 different pathogens, individual ticks.
And Dr. Stafford, in terms of how often are ticks that are sampled in Connecticut found to have more than one pathogen? Do you know?
It's about the same rate. So you have just a few percentage that will have, say Borrelia and Babesia or Borrelia and Anaplasma. And then there's that small percentage, again, similar to what Dr. Rich has found, around that 2-3%, or even actually less than that. On occasion, we'll get all 3 pathogens present in one tick. So theoretically, you could get, say, Lyme disease and babesiosis at the same time, either from a single tick bite or from 2 different, tick bites that occurred around the same time. So sometimes that can complicate the, clinical presentation. And so clinicians should always bear in mind that even though Lyme disease is the major tick-borne illness that we're dealing with here in New England, we do see tens to a few hundred cases here in Connecticut anyway of the other black-legged tick associated diseases as well. So that should also be part of the differential diagnosis.
Okay, well, thank you. Unfortunately, we do have a time limit, and this concludes our webinar. I'd like to again thank each of the panelists for sharing their knowledge, and I'd also like to thank each of the pesticide state lead agencies for their support and participation. We apologize for any questions that we weren't able to answer, but feel free to contact panelists separately or contact EPA, and we'll do our best to provide additional information. And thanks for participating, and we're hoping you'll be able to join us for our follow-up webinar on mosquitoes on July 16th. Thank you.